Hello students, today we will learn about biotechnology, society, risk, ethics and patenting. So let's begin. Advances in biotechnology and their applications are most frequently used associated with controversies. Based on their perception to biotechnology, the people may be grouped into three broad categories. First is strong proponents, and third is neutral group. Strong opponent who oppose the new technology as it will give rise to problems, issues and concerns humans have never faced before. They consider biotechnology as an unnatural manipulative technology. Second is strong opponents, that means who consider that the biotechnology will provide untold benefits to society. They argue that for the centuries the societies has safely used the products and processes of biotechnology. Third point is the neutral group. Neutral group, what happens? Neutral group of people who have a balanced approach to biotechnology. This group believes that research on biotechnology with regulatory systems and extending its fruits to the society should be pursued with a cautious approach. The risk and benefits of the developments of biotechnology may not be much different from that of any other branch of social science. Now let's know about benefits of biotechnology. The fruits of biotechnology are beneficial to the fields of healthcare, agriculture, food production, manufacture of industrial enzymes, and appropriate environmental management. It is a fact that modern biotechnology in various forms is woven tightly into the fabric of our lives. Our day to day life is inseparable from technology. Imagine life about 1 to 2 centuries ago where there was no electricity, no running water, sewage in the streets, unpredictable food supply and expected lifespan of less than 40 years. Undoubtedly, technology has largely contributed to the present day world we live in. Many people consider biotechnology as the technology that will improve the quality of life in every country, besides the maintaining living standards at a reasonably higher level. Now let's know about ELSI of biotechnology. ELSI of biotechnology. Why so much uproar and negativity to biotechnology? Again, starting from ELSI of biotechnology. Why so much uproar and negativity to biotechnology? This is mainly because the major part of the modern biotechnology deals with the genetic manipulations. These unnatural genetic manipulations, as many people fear, may lead to unknown consequences. ELSI is the short form of ethical, legal, and social implications of biotechnology. ELSI broadly covers the relationship between biotechnology and society with particular reference to ethical and legal aspects. The probable positive and negative effects of biotechnology, particularly in the developing countries, we will know now. Positive effects. What are the positive effects of biotechnology? Improved health and lifespan through more effective pharmaceuticals and vaccines, etc. Increased crop production and more food. Next point is decreased dependence on import of food, fertilizers and chemicals, enhanced biomass production for energy. Next point, use of plants and animals as bioreactors. Next, improved food storage facilities. Next point is increased production and health of livestock. Now let's know about negative effects. More dependence on developed countries and private companies for technology and resources. First point. Second point, increase in unemployment as the labor requirement is less. Third is reduction in natural biodiversity and natural ecosystems. Fourth point is more legal and financial complications. Fifth point is increased growing of cash crops at the cost of food crops. Next point is production of more and more herbicide resistant plants. Now let's know about risk and ethics of biotechnology. The modern biotechnology deals with genetic manipulations of viruses, bacteria, plants, animals, fish and birds. Introduction of foreign genes into various organisms raises concerns about the safety ethics and foreseen consequences. Some of the popular phrases used in the media while referring to experiments on recombinant DNA technology, now we will discuss. First point is manipulation of life. Second is playing with God. Third is man-made evolution. The major apprehension of genetic engineering is that through recombinant DNA experiments, unique microorganisms or viruses either inadvertently or sometimes deliberately for the purpose of war may be developed that would cause epidemics and environmental catastrophes. Due to these fears, the regulatory guidelines for research dealing with DNA manipulation were very stringent in the early years. So far, risk assessment studies have failed to demonstrate any hazardous properties acquired by host cells or organisms due to transfer of DNA. Thus, the fear of genetic manipulations may be unfolded to a large extent. Consequently, there has been some relaxation in the regulatory guidelines for recombinant DNA research, and it is now widely accepted that biotechnology is certainly beneficial to humans, but it should not cause problems of safety to people and environment and create unacceptable social, moral, and ethical issues. The public fears of biotechnology, besides some of the risk, social, and ethical issues, can be better understood with special reference to the some points to some extents. First is therapeutic products for use in healthcare. Second is genetic modifications of foods and food ingredients and their consumption. Third is release of genetically engineered organisms into the environment. Fourth is applications of human genetic research. Now let's know about recombinant therapeutic products for human healthcare. It is fortunate that there is no serious criticism about the use of recombinant products for medical applications. This is mostly because the therapeutic products and strategies are designed to cure diseases, alleviate sufferings, and improve the quality of life. Further, the products are used quality under the medical supervision. In general, the recombinant products designed for human healthcare are more readily acceptable by the public. Good examples are the use of insulin, interferons, tissue plasminogen, activator, and various vaccines. Now let's know about genetic modifications and food consumptions. The overall objectives of genetic modification with reference to foods, now we will discuss. First point is to increase the quality and quantity of existing foods. Second is to produce new products. Third is to improve the financial returns. Each country has its own regulations for introducing foods into the market. For instance, in USA, the Food and Drug Administration, that is in short FDA, regulates the introduction of foods, drugs, pharmaceuticals and medical devices into the marketplace. As regards the foods and food ingredients developed by genetic engineering, the FDA believes that no new regulations are needed. The existing regulations for the assessment of foods for safety by toxicity, allergenicity, and impurity testing and are adequate. If the chemical composition of the existing food is altered by genetic modification, it should be specified and the new product should be accordingly labeled. To highlight the public perception of genetically modified foods, some selected examples of food ingredients and their acceptance or certain controversies related to their use we will now discuss. First is chymosin. Chymosin is milk clotting proteolytic enzyme that hydrolyzes the milk protein casein to produce curd, which in turn to process into cheese. Traditionally, chymosin is derived from the stomach of calves in the form of rennet. By genetic engineering techniques, the chymosin gene was cloned and expressed in E. coli. This resulted in a large-scale and cost-effective production of chymosin. The chemical composition, structure, and biological activity of recombinant chymosin were identified to the chymosin of rennet. FDA gave license to chymosin for its commercial use and it is now widely used in cheese making. Now let's know about tryptophan. In 1989 to 90, an unusually high incidence of the disease eosinophilia myalgia syndrome, in short EMS, was reported in USA. EMS is a rare disease with muscular pain and respiratory complications and may be fatal. Investigations revealed that the victims of EMS were consuming large quantities of food supplement tryptophan, that is tryptophan. This tryptophan was produced by genetically engineered microorganisms. Chemical analysis of the commercial preparations revealed the presence of certain metabolic derivatives of tryptophan. The most important among them was 1,2,1-methylene bis, that is tryptophan, which was responsible for EMS based on the result of animal experiments. It was concluded that the pharmaceutical companies did not take adequate care for purification of tryptophan. Consequently, the recombinant tryptophan, even without the impurities, was banned for human consumption in USA. Now, let's know about bovine somatotropin story. Bovine somatotropin, when injected to dairy cattle, increases the milk production significantly. By recombinant DNA technology, the gene for BST means the bovine somatotropin story, was cloned and expressed in E. coli. The recombinant BST, or in short RBST, so produced when injected to cows, was found to increase milk production by 20 to 25%. The effect of RBST was considered from two aspects on animals and consumers. For let's know about effect of RBST on animals. Administration of RBST can produce localized swelling at the site of injection. There may be some other adverse effects like increased susceptibility to infection, decreased reproductive capability. The proponent of RBST argue that this problem could occur even in normal animals. Second is effect on human health. The natural BST or even RBST increases the body levels of insulin like growth factor 1, in short IGF-1, which in turn enhances milk production. There is evidence that IGF-1 stimulates growth of cancer cells. This causes concern among the consumers of milk produced by using RBST. The counter argument is that RBST injection after about 100 days of lactation begins is not associated with increased level of IGF-1. The opponents of RBST strongly feel that Since milk is consumed by most people, any inherent risk, however small, is unacceptable. Recombinant BST was licensed in USA for use in dairy cattle in 1994. Some countries, in fact, have banned the use of RBST. There are some people, particularly among the scientists, who believe that the hue and cry raised against RBST is more due to economic and political reasons. It is feared that by RBST use, the dairy industry may be controlled by a large industrial group and the small dairy farms may become unprofitable. The RBST story is an interesting illustration of the problems surrounding the use of genetic engineering. Now let's know about recombinant foods and religious beliefs. For some of the ethical concern of the use of recombinant foods are related to religious beliefs besides food habits. For instance, first point is transfer of pig genes into sheep may offend the sentiments of Jews and Muslims. Introduction. Second point is introduction of animal genes into food plants may invite opposition by strict vegetarians. Third point is transfer of human genes to food animals may be unacceptable to some people. Fourth point is feeding of human gene concerning organisms to animals sounds in bad taste. At present, the genetically modified yeast that produce recombinant proteins after their use are fed to animals. The religious groups in general are selective about the foods to be eaten. However, they are not so rigid when it comes to use of medically derived products. For instance, Jews and Muslims may accept pig derived insulin for use in diabetes patients. This is due to the fact that all religious faiths consider human life is the most valuable and its preservation the first priority. Further, the general belief is that the human body is violated only by oral consumption and not by injection or surgical interventions. Now, let's know about eating genes every day. Every day, we eat plants and animals and various products derived from them, besides a large number of bacteria. In other words, we regularly consume genetic material, the DNA, organized into genes in various organisms. So far, no one has attempted to categorize genes as vegetarian and non vegetarian as we do for foods. Now, let's know about, are genetically modified foods safe? In short, are GM foods safe? The production of transgenic plants and animals by genetic engineering techniques has now become routine. These organisms will enter the food chain in the form of genetically modified foods, and some social and environmental groups are against the consumption of genetically modified foods. These people insist that the genetically modified foods should be specifically labeled. As regards the safety of genetically modified foods, opinions range from one extreme that they are absolutely safe and improve human nutrition to the other that they should not be consumed at all. Most of the people have opinions somewhere between these two extremes. Now let's know about release of genetically engineered organisms. The release of genetically engineered organisms, also called as genetically manipulated organisms, into the environment has been a controversial issue and continues to be so. It is feared that the release of genetically engineered organisms into the environment could have far reaching consequences. This is due to the fact that the living genetically engineered organisms proliferate, persist, disperse, and sometimes may transfer their DNA into other organisms. It is further feared that there exists a possibility of genetically engineered organisms displacing the existing organisms besides creating new species. They may lead to severe environmental damage. For the reasons stated till now, we studied the regulatory authorities are very careful in permitting the field trial of genetically engineered organisms. Further, the release of genetically engineered organisms into the environment has to be carefully monitored and recorded. Now, let's know about ICE minus Pseudomonas syringe. A genetically modified strain of Pseudomonas syringe was the first genetically engineered organism that was given permission for field trials in 1987. This organism is a genetically engineered ice-10. When sprayed onto the leaves could prevent frost damage to the plants. Now let's know about field trials with other genetically modified organism. A number of open field trials have been conducted with several genetically engineered organisms during the past 2 decades or so. The studies concluded that the genetically modified microorganisms do not persist in the environment for long, do not transfer the genes into other organisms, and do not exhibit any abnormal biological functions. Thus, the initial apprehensions on the use of genetically engineered organisms appear to be unfounded. Now let's know about release of transgenic plants and animals. The transgenic plants developed for higher quality and quantity of foods in general are more liberally permitted to go to fields. It is generally believed that the transgenic plants do not significantly differ from the natural cultivars obtained by plant breeding experiments. The transgenic Bt plants as a cotton, corn, soybean, potato were approved for cultivation in USA. However, some countries did not allow Bt plants, Bt means Bacillus thuringiensis plants in their fields, and Bt rice was not allowed in Philippines. Bt cotton is not allowed in France. The transgenic animals have not posed as big a problem as the transgenic plants. This is due to the fact that animals can be much easily identified and contained since copulation in is animals can be done as derived, unlike in plants where pollination is difficult to control. A majority of transgenic animals are used for medical purposes, hence they are generally appreciated, but the major problem for transgenic animals comes from animal activities. Many people and also some governments are suspicious for the use of genetically engineered organisms due to various reasons, risk, societal beliefs, and economic concerns. Now let's know about biological warfare. The most serious hazard associated with genetic engineering is the construction of harmful biological agents, that is viruses, microorganisms, either deliberately or otherwise. However, so far there have been no records of any new infectious agents created by recombinant DNA technologies. Most of the countries of the world are signatories to the Biological Weapons Convention of 1972. As a signatory, a nation please never to produce microbial or other biological agents or toxins, whatever may be their method of production for use in wars. Many people are, however, concerned about the possible use of gene manipulations for military purposes. Now, let's know about applications of human genetic rDNA research. The ultimate goal of advances in biotechnology is for the benefit of mankind, either direct or indirect. Biotechnology largely contributes to human genetic research involving some areas. Let's know about the first is genetic testing and screening for diseases, genetic portfolios. Third is human gene therapy. Let's know about this. First is genetic testing and screening. The techniques are now available for prenatal testing to specifically detect whether a fetus carries genetic defects or not. This will help the parents to be better prepared for the future baby. The negative aspect of parental testing is that the couple may opt for abortion even for a minor genetic defect or sometimes for gender biases. Now let's know about genetic portfolios. The elucidation of the entire human genome sequence and identification of genes has now become a reality. It may soon be possible to have individual genetic portfolios that will diagnose future health complications, example, risk for cancer, heart disease, etc. The genetic portfolios based on the genes will foretell an individual's future, which is now being predicted through stars, that is astrology. Genetic portfolios of individual may pose certain problems with regard to marriages insurances. Who would to be a spouse of someone who will soon be a victim of cancer or heart attack? Which insurance company would insure a person with a very high risk of diseases? Many ethical committees are of the opinion that insurance companies should not require or should not be allowed to have access to individuals' genetic portfolios. Now, let's know about human gene therapy. Theoretically, correction of genetic defects is possible by gene therapy. The present status of human gene therapy has now we will discuss. Now already discussed. Okay. From the ethical perspective, gene therapy involving introduction of genes into a patient is comparable to the practice of transplantation of heart organs like heart, liver, lungs, etc. Therefore, there is not much controversy over gene therapy as long as it is intended to be used to alleviate serious medical disorders. However, the gene therapy must be under a close supervision to satisfy medical, legal, ethical, and safety complications besides addressing the public concerns. Now let's know about germline gene therapy. This is not being carried out at present due to technical, ethical, and social reasons. Manipulations of germ cells will lead to serious problems and complications. At one international meeting on biotechnology, the following was the final message given to biotechnology company on the applications of genetic engineering to humans. Provide the information and listen to the public. Human embryonic stem cell research. Let's know about human embryonic stem cell research. The human embryonic stem cell, in short ESC, lines were established in November 1998. These cells are capable of giving rise to any human cell type. ESC lines open the possibilities of treating diseases with cell therapy. Disorders that involve the loss of normal cells such as diabetes mellitus Parkinson's disease, Alzheimer's disease could perhaps be corrected with cell therapy. This may, however, take more time. There are many ethical and legal issues involved in ESC research, besides several objections raised by the public. In fact, the US federal government has banned the use of federal funds for human embryo research for over 20 years. At present, most of the research on ESC is being supported by private companies. Now, let's know about cloning humans. Human clonings. Okay, after the cloning of the sheep Dolly in February 1997, some groups of researchers naturally became interested in to explore the possibilities of cloning in humans. The very thought of human cloning has become a highly charged and controversial issue. As such, considering the considering the biomedical ethics, most of the countries have banned research related to human cloning. It can be predicted at present whether someday human cloning may become inevitable. Scientists who were awarded Britain's first license for human cloning have recently in 2005 created an early stage of human cloned embryo by using nuclear transfer. The researchers such as are not interested to make babies by cloning. Instead, they wish to create test tube embryos to supply the stem cells that can give rise to every tissue in the body. By this approach, it might be one day possible to repair tissue damages and cure many diseases like Parkinson's disease and diabetes mellitus. Now, let's know about patenting biotechnology inventions. Biotechnology involves the production of a large number of commercial products of economic importance. Broadly, biotechnology invention can be categorized into two forms. First is product and second is processes. First, biotechnology products. Let's know about the biotechnological products. First is living entities. The various forms of life existing and life supporting systems derived from the living organisms are regarded as living entities. These include microorganisms, animals and plants, cell lines, organelle and plasmids and genes. Second is naturally occurring products, that is the primary and secondary metabolites produced by living systems, like example alcohol and antibodies. Now let's know about biotechnological processes. Biotechnology involves development of several processes like isolation, purification, cultivation, bioconversion, etc. Novel, innovative, simple and cost-effective processes are developed for the isolation and creation of these products. Let's know about some examples. First example is production of monoclonal antibodies. Second is bioconversion of sugar to alcohol. Third is microbial production of antibiotics. Now let's know about what is a patent. What is a patent? A patent is a government or patent office issued document that allows the holder, that means patentee, the exclusive right to manufacture, use or sell an invention for a defined period, usually 20 years. The patent may be regarded as a legal document safeguarding the privileges and rights of an invention or inventor. The very purpose of patenting in biotechnology is to ensure the just financial returns for those who have invested heavily. The finances intellectual abilities besides the hard work. Now let's know about intellectual property rights. Patent is the most important form of intellectual property rights, that is in short IPRs in biotechnology. The other IPR are trade secrets, copyrights, and trademarks. Now let's know about trade secrets. The private information pertaining to specific formulations and technical procedures that a company wishes to protect from others are regarded as trade secrets. The best known trade secret is the formula for Coca-Cola. Only 5% in the world are said to know the formula, which has been kept in a bank at Atlanta, Georgia. The trade secret of Coca-Cola has remained a secret for more than 100 years. Copyrights. Let's know about the copyrights. The protection of authorships of published work comes under copyrights of IPRs. Now let's know about trademarks. The specific symbols in words to identify a particular product or process of a company constitute trademarks. Now let's know about the process of patenting. As regards patenting of biotechnology inventions, the products of process are patentable. For patenting, the following criteria must be satisfied. First is the invention must be novel. Second is it must be useful. Third is the patent application. The patent application must provide the full description of the invention. In contrast to various other technological inventions, biotechnology inventions most often relate to living materials. For this reason, the patenting of biotechnology inventions is often associated with difficulties. Patenting in general is a commercial decision assisted by legal advice. The patent application must be prepared by an expert with the assistance of a patent lawyer. It must contain the detailed information about the invention, background, description, explaining the nature of the invention with figures and illustrations and its utilities. The patent office, after a thorough scrutiny of the application, may reject or grant the patent. If rejected, the application may appeal to the Patent Appeals Board. If again rejected, the decision may be challenged legally. Many a times, the process of patenting becomes a frustrating experience. Thomas Edison, probably the only individual in the world who, with more than 1000 patents to his credit, once said about patenting an invitation to a lawsuit. Now let's know about patenting in different countries. Patenting laws are very complex and vary between different countries. It is interesting to note that a patent application rejected in one country may be given patent in other country. The best example is the production of recombination human tissue plasminogen activator, in short TPA, in E. coli. Genentech company was awarded patent for TPA production in USA, while it was first rejected in UK after appeals and legal battles. Patent was finally granted in UK also. Now let's know about patenting microorganisms. A genetically engineered strain of the bacterium Pseudomonas called superbug was the first microorganisms to be patented. Superbug is capable of breaking down the multiple impurities of crude oil. It took almost 10 years of procedural delays and legal battles to get the superbug patented. Now many microorganisms are patented. Now let's know about Patenting multicellular organisms. A genetically manipulated mouse, namely the onco mouse, was the first and probably to date the only animal to be patented in both USA and UK. This transgenic mouse carries a gene that makes it susceptible to tumor formation. Now let's know about patenting genes. The Human Genome Project, in short HGPS, have elucidated almost the entire sequence of human genome besides identifying the genes. Patenting of the DNA sequences and genes has become a controversial and issue. Debatable issue. Some researchers and institutions, in fact, have approached the patent offices for grant of patents. So far, patents have not been granted for issues for genes. The most important reason being and that the genes are natural, universal and inherent biological functional units of all individuals, presenting and cell or tissue donors' rights. While dealing with human tissues or cells, the involvement of the donor of source material becomes very important. John Murray was a victim of hairy cell leukemia, a rare form of cancer. His spleen was removed and MO cell lines developed. These cell lines were patented by the researchers and their associated organizations. Murray, however, was not involved in the patenting. Murray filed a suit in the court on the ground that he should also be a party to the profits derived by using his tissue. The court allowed his claim to share the profits. Now let's know about plant breeders' rights. Agriculture for the first time was included in 1994 in the Trade Related Intellectual Property Rights and TRIP, in short Trade Related Intellectual Property Rights, that is in short TRIPS. TRIPS is a major concern for developing countries. The plant varies in many countries, not in India, are protected through plant breeders' rights, in short PBR, or plant variety rights, PVR. PVR provides legal protection to the original breeder or owner of the plant variety. It is believed that PBR will encourage innovative research and plant breeding programs in view of the expected financial returns. Plant breeders' rights are capable to the patent rights. Under TRIPS agreement, plant breeder possesses the exclusive rights over the plant developed. It prevents the third parties from using the plant without the owner's consent. Although PBR will increase competition and development of good plant varieties, the owner's motto would be profit making. Therefore, PBR will prohibit the free exchange of plant materials besides threatening the farmers' rights. It is feared that PBR will benefit rich farmers and developed countries only. Now let's know about patenting and biotechnology research. In the earlier years, research used to be mostly for academic interest and worthwhile scientific contributions, and researchers were purely academic oriented with not much interest in financial gains from what they do. Further, unlike now, the research used to be supported mostly by governments or universities. There used to be no secrecy in research. Academic recognition and outstanding research publications open to all were adequate to satisfy the researchers. Watson and Crick, who discovered DNA structure, never had a thought of patenting. The situation has now changed. A good proportion of research is either being conducted or financially supported by private companies or universities. It is quite natural for these companies to expect returns for their investments. There is a lot of change in the altitude of researchers also. For many scientists, financial gains have become more important than academic recognitions. Consequently, some people carry out research secretly and opt for patenting of their discoveries rather than publishing. We have to accept the fact that the progress of research is usually much faster in private companies compared to many government organizations. This may be due to the higher financial returns and efficient administration. Probably the biotechnological research might not have progressive progressed to the same extent as it is today without the dreams of getting the patients by scientists and research funding organization. Now let's know about biotechnology and the developing countries. The major proportion of research related to biotechnology is carried out by the developing countries. The fruits of biotechnology are probably more useful and relevant to developing countries. Let's know about its application. First is increased level of nutrition with improved nutrition composition in the foods through transgenic plants. Second is prevention of child deaths by appropriate immunization using recombinant vaccines. Third is supply of clean drinking water and improved sewage disposal by appropriate biotechnological treatments. Despite the known benefits, some of the developing countries are reluctant to open doors for the advances made in biotechnology. This may be more due to political considerations rather than economic reasons. The applications of biotechnology, particularly to human healthcare, may someday be regarded as a barometer to evaluate the progress of a nation. Finally finished. Any questions regarding this? You can ask me in the comment box. And this knowledge is for biotechnology students who are preparing biotechnology and also studying biotechnology, for BSc biotechnology, MSc biotechnology, and who are preparing CSIR UGC net also can refer to my channel and subscribe my channel to know more updates. Thank you.